It's a real pleasure today, at risk section, by introducing our keynote speaker, Ange Rasmussen. Ange is a vaccinologist and scientist based at the University of Saskatchewan in Canada. She's a US citizen, and she's a co-editor of Vaccine Journal and a staunch online vaccine advocate. Ange, thanks very much.
Oh, they do? Oh. Okay, so I could have given the talk without the microphone. I totally changed my talk, and I made this talk like 3 different times because I really wanted to be, I don't actually work on the health effects of vaccines, except I actually do because I study the host response to vaccines. And I usually study that in terms of overall transcriptomics, and yesterday, I heard everybody almost say that they were very interested in sex biases. And that happens to be one thing that my lab does work on a lot. We don't actually do that much vaccine research, so I'm kind of a poser vaccinologist, I have to say. I have learned so much about the vaccine field, but my role in my lab focuses mostly on pathogenesis studies and developing animal models for that, and then interrogating the global response to infection or vaccination with transcriptomics primarily. So let's just get right into it. I tried to come up with a catchy title, and I bolded sex to really emphasize the point that I'm trying to be responsive to everybody's interests. This is how my lab approaches this problem. We start with our experimental models in the laboratory. Occasionally we have some samples from the field or the clinic, but mostly our bread and butter is mouse and monkey models. We do standard virology, biochemistry, bench science, that sort of thing, and then we take samples, we put it through our omics pipeline. Typically that's transcriptomics. Sometimes we do proteomics and metabolomics though too. This results in a ton of data, and it usually ends up, you model it, and it turns into a hairball like this thing. And that's hard to make sense out of, but we have a lot of really new techniques, and we have AI now, which enables a lot more modeling than we could do before, just because it makes it so much more accessible to so many people, including myself, because I'm also a poser systems biologist, and until I had Claude Code, I needed a bioinformatician to do everything for me except for the functional analysis, and now I can do the pipeline too. But what we get out of this is a lot of different hits, basically, to either generate new hypotheses, repeat the cycle all over again, or we can do things that have more clinical applications, like biomarker panels, novel drug targets, and then, of course, vaccine correlates of protection. You can also look at the overall response to vaccination using this method. And then you just repeat it. And I've found that it is a really great way to develop a model and then start interrogating it for what's going on in terms of pathogenesis. So I tend to favor the host because everybody in the virology field tends to focus on the virus, and they like the mutations, and they like all the different proteins and seeing what they can do. But in my experience, the host is just as, if not more important in determining the outcome. And I'm going to show a little bit of data later where we've used machine learning to predict outcomes for Ebola virus. So I'm going to go through this really quickly because I know I only have a half hour, and I don't like being the person who's going way over, but I'll probably go over a little bit, and it seems like this conference is cool with that. But I'll try to be good. Like I said, a lot of the models I use are mice and monkeys. Mice are great because, especially when you're working with a pathogen that requires high containment, because they're easy to handle, they're reproducible, they're fairly cheap, they're very homozygous, so it's fantastic for generating reproducible data most of the time, but that comes with a cost because they don't have very much genetic diversity, they're not comparable to human populations, and oftentimes it requires human pathogen adaptation to the rodent model, which used to be not much of a problem. I'm going to talk in a little bit at the end of my talk about how changes in US policy are affecting researchers all over the world, and specifically how they would affect my research. So alternative to mice is non-human primates, and they are genetically much closer to people. They're highly heterozygous, so they're more comparable to a genetically diverse population. Most of the time you can infect them with the wild type pathogens. They don't always develop the same outcome, but you can usually infect them. But they're not genotyped. Depending on the species, sometimes the genome might not even be in great shape. They're very expensive now and very hard to get after the pandemic. There's limited reagents, and they're big. And in containment, like, that can be a real problem because they have claws and teeth, and they're smart, and they do not like being in a containment lab. So both of them have their drawbacks. There's a lot of challenges in modeling emerging viruses. Every animal model is useful but imperfect. And for Ebola virus, wild-type Ebola does grow in mice. They are susceptible to it, but it really doesn't replicate very much. It doesn't cause disease. It doesn't kill them. Mouse-adapted Ebola was made about 25 years ago at USAMRIID. It's the basis for adaptation is mutations in the MP and VP24 proteins after 9 serial passages through progressively older suckling mice. And what happened is these animals die. Ebola infects the target organs, the liver and the spleen, and the secondary lymphoid organs. They lose weight, they get sick, but they don't have any of the coagulation abnormalities that we see in Ebola virus disease. So we wanted to know if increasing the host genetic diversity in the mouse model would result in an expanded range of host responses. And this is the model that I've primarily used to look at these sex biases, and that's the collaborative cross, which is an 8-way cross between 5 inbred lab-derived strains and 3 wild strains. And so this covers 90% of the genetic diversity across the entire mus musculus species and includes these 2 wild-derived subspecies of mus musculus. So the average lab mouse has about 10 to 15% of the genetic diversity represented. So this represents a huge expansion of that. And this is how they were made. They take these 8 founder strains, they cross them together again, and then they back cross them for 15 to 40 generations until they're mostly homozygous. Each collaborative cross genome will be a mosaic of the 8 founders, and they make different lines by shuffling up the order that the founders are crossed in at the top. So now there's about 200 of these that you can buy. Their genomes are, at this point, I think they're all completely sequenced. They used to have them genotyped and they would impute them, but now you can get the whole sequence, and it has really made a big difference for some of the viruses that I work on, because you do have things like some mice with MX1, for example, where they don't have a functional MX1, much like traditional lab mice. There are others that do have a functional MX1. So you can actually, by looking at the genomes, look at different host genes and their responses and how they impact that. We also can cross these together to make what we call CC-RIX, or recombinant inbred intercrossed, and they have heterozygosity consistent with or greater than human populations. So if you take a regular CC line and cross it together, it's equivalent essentially to an inbred mouse. If you take 2 of them and cross them together, then it's essentially equivalent to a human in terms of the diversity. So we put mouse-adapted Ebola into these mice, and we got really lucky on our first time. We got 2 completely, extremely opposite phenotypes. One was lethal with hemorrhagic syndrome, and anybody who's ever done any mouse work knows that this is not a normal-looking mouse liver. It was described to me by Atsu, my colleague who did this work in the BSL-4 at Rocky Mountain Labs, as the texture of soft tofu. And it's hard to see in this picture, but there's actually little spots of blood welling up on the surface of the organ, because these animals have their coagulation times extended within 24 hours of death, and their serum fibrinogen plummets over the course of infection. We also have a strain that is what we call tolerant. So these animals are infected. However, they don't have any obvious morphology of disease. They get a little bit sick, and then they recover fully. They both get infected, as I said. These are sections from the liver staining for VP40, the viral matrix protein. The lethal with hemorrhagic syndrome animal had much more virus in the liver, punctate infections in the tolerant animals, and we think that this is because in the tolerant lines, the host response is controlling the infection and clearing it, whereas in the lethal ones, it goes out of control. So we've repeated this a lot of times. That was with CC-RIX mice. This is with 10 lines of CC mice, and we see the same thing almost every time. The lethal ones all die within a different time frame sometimes, but they all do die. The tolerant ones all lose a little bit of weight at the same clip, and then they recover. Viral replication, we see totally comparable amounts of viral RNA but we do see a 1 to 2 log difference in terms of the amount of infectious virus that's being produced. So we think that this is probably the host response controlling, again, the infection and preventing the virus in the tolerant animals from infecting as robustly in those animals compared to the ones with lethal syndrome. So we've done this with 75 different collaborative cross backgrounds, and we see the same thing in the CC and the CC-RIX. About a third of each have the tolerant phenotype, the lethal with no hemorrhage phenotype, which is what we see in traditional lab mice, and then we have the lethal hemorrhagic phenotype, and again, it's about a third of each. So when we did transcriptomics on this, we saw some pretty big striking differences. And this is primarily, there's a huge suppression in both the spleen and the liver of antiviral genes, of interferon-related genes. And this eventually goes up. And this was something, when I first started working on Ebola, that drove me crazy. Because at day 5, when these animals are dying, their interferon everything is just way up. And that doesn't make sense to me, because these animals are about to die from Ebola virus. And I think the reason for that is this early suppression allows the virus to take off, and then the host tries to catch up by day 5, and by then it's too late. Whereas with the tolerant animals, they actually upregulate this on day 1, and then they have a more controlled response. And so what we think is that the virus is the same for all of them. They're all infected with the same dose, but the response is very different, and this early control of the antiviral response is critical for these animals to survive Ebola virus infection. So we wanted to know if sex influenced it, and it was primarily because after the 2014-2016 outbreak, there was this observation that there were female patients who were significantly less likely to die than male patients. And that can change depending on if the female patient is pregnant or not. But this did remain significant after all these adjustments by age, and even when they restricted the analysis to just PCR-confirmed Ebola cases. So this was a lot of patients, and it looked like there was a big sex bias, so we wanted to see if we could see that in the collaborative cross. And Jim, this is what I was bragging about when I was walking in. We have these really extreme sex-biased phenotypes for 2 of these lines. So in this one, all the females survive. In this one, all the males survive. And we've done so many. We use this as a control for tolerance in males. We have done probably 40 or 50 of these mice, and the males always survive. The females, most of the time, die. So this is a really great model for studying this. The only problem, which I'll get to later, is that it's getting to be very, very difficult to work on viruses like Ebola in any containment lab in any country. There is so much more scrutiny on virology research that it is becoming very difficult to do this type of work. But we also see other sex-biased phenotypes within the collaborative cross too for Ebola. They're less extreme, but we do see differences in terms of virus production, in terms of the amount of time that it takes, the coagulation times before the animals die for the ones that have lethal with hemorrhagic syndrome, and we see differences in weight loss as well between sexes. So here's the full list of what we've screened so far. As you can see, we actually have severity biases going both ways. So hopefully in the future, what we'd like to do, assuming that we can actually do these studies, Vito is in the process of upgrading to BSL-4 containment, but that process is always much longer. It was supposed to go live in 2025, I think. Now it's 2028. In any case, we will be able to actually look at these biases, and I'm doing a little bit of work now that I don't have any data for yet, trying to look at the roles of VP35 and VP24, which are the interferon antagonists, in the presence of different sex hormones. So we can do that at BSL-2. We also can distinguish these male and female animals by their transcriptional profiles, and we haven't done this in the lines that have the really extreme phenotypes because, unfortunately, the COVID pandemic happened and that shut down a lot of this work. But we have actually seen transcriptomic differences in lines without a sex bias or with a more subtle sex bias when we have done both males and females. For reasons that are not really scientific, we were funded to do some of this work by DARPA, so they wanted only male mice. And I argued with that, considering it's not just men that serve in the military, but they were insistent that we only do males, so that's why I have a disproportionate number of males. But we see this difference as well in these cynomolgus macaques. And this is a much larger study that we did with 60 different animals at the BSL-4 in Galveston. And we're still analyzing this data because, again, the pandemic got in the way. But we actually have differential outcomes for this, too, because we infected them with different doses via different routes. And very interestingly, when you infect with the conjunctival route, it requires 10,000 PFUs of Ebola delivered to each eye. And if you infect with the aerosol route, it takes 0.1 PFU to get uniform lethality. There's no such thing as 0.1 PFU, but Ebola virions are very long and thready, and they probably aggregate. So that's why we think that that data is like it is, but it's a really striking model. So we will be able to actually compare some of our mouse data to this non-human primate data going forward. Oops, that slide was supposed to be at the end. Okay, so the next thing I'm going to talk about briefly is just the role of sex hormones in pathogenesis. Sabra recognizes these. They're her papers, I think. One of them is. So there are different effects when you treat animals or you look at sex hormones during different kinds of infections. And so we wanted to see if this was the case also with our Ebola data. And indeed, we see that tolerance is increased with an upregulation in these sex hormone biosynthesis pathways. So we wanted to look at this also in some of our other models. And the nice thing about working on the host is that I don't have to pick a virus. I can pick any emerging virus, and that's why I work on as many as I can, because they all scare me. So this is a study we did also with this DARPA program to look at MERS coronavirus severity. Now, MERS requires a transgenic mouse. So we have a dose-dependent mouse model in the HDPP4 transgenic model that we use to look and compare tolerant lethal. And I'll show a little bit about this that Maren has done in my lab. We also have another study that we need to finish analyzing, which is a lot more complicated because it involves comparing marmosets, which succumb to lethal disease, or they always meet endpoint criteria for humane euthanasia, and alpacas, which are completely tolerant. They have no clinical symptoms whatsoever, and they shed a ton of MERS. So we compared these things, and it's actually a little bit different than Ebola. There's actually an early suppression in the lungs in the MERS model in the animals that are tolerant compared to the lethal model. So we've been looking into that more closely. This is just basically showing the data, and what I just showed, you know, we have this tolerant versus lethal in this dose-dependent model. It's not really a model of tolerance because it's a dose model, but it served its purpose for the study. And we see, as many people have in this transgenic model, just something to look at is that there is this brain involvement usually, and that's not typical of MERS. This is a problem with most of the coronavirus mouse models in the sense that if the transgene for the receptor is overexpressed in the CNS, you will get encephalitis sometimes, which is not typical of MERS. In this case, only the lethal animals, one of the tolerant, or a couple of the tolerant animals had it at day 5, but for the most part, we did not see replication in the brain in the tolerant animals. So lethal versus tolerant host responses, again, were like Ebola, but not like Ebola in the sense that the tolerant animals suppressed a lot of these responses early on, whereas the lethal animals upregulated them. This is an upstream analysis, so this is looking at our genes and trying to predict which upstream regulator that may or may not be transcriptionally regulated would be doing, basically using our gene profiles to explain that. This is just a traditional canonical pathway enrichment, but we see the same pattern for both, in that the tolerant animals are really suppressed, and they actually don't have much of a response at all. That may be, again, due to the fact that this is a dose-dependent model, and some of the animals aren't getting really productively infected, but we do see this really striking difference. So we wanted to look at hormone-dependent responses because we were really interested in this, and Maren, in particular, is going to be so sad that she's not here because she really wanted to meet Sabra at some point. So we'll have to make that happen. But we started looking at all of these different hormone pathways. Obviously, you can't really look at hormones with transcriptomics because there's not mRNA for them, but you can look at the receptors, and you can use this upstream analysis to predict whether or not they would be involved. And so, I mean, this was very interesting. We saw these very different responses in the females versus the males, where early on in the females, we saw, this is all in lethal animals, by the way, we just saw all of these interferon genes come up, and then we see all of what Marin has called proviral genes in the calcium signaling pathway not really up that much. By day 2, it's the opposite for the males, but by day 4, it has changed, and the females are now upregulating this too. So we saw this difference in the time. The study wasn't designed to look at sex bias, but we actually were able to find some of the animals' sexes from these groups. They weren't recorded by mouse, but we knew how many there were, and we knew which one was male and which one was female. We just didn't know which male or female was which. And we found that in the tolerant group, some of the animals died. They were all male. So we wanted to look at that. We looked at these sex-specific transcriptional responses, and again, this study wasn't designed for this, so this was about all we could do with it. But we did, again, see these fairly striking differences, particularly in the tolerant animals, where the females have almost no response. They seem to be almost resistant to it. I mean, they do have a little bit of virus, and they have a little bit of a response, but not much of one, whereas the males have this big response on day 1, not much of a response after that, and then they die. So we want to look into this further. We haven't been able to do these studies in terms of just funding, because doing these studies requires gonadectomization and hormone implants, and it's a lot to put in there without any grant funding for it. So that's something we're working on right now. We've also done this with COVID. So early on, this was way early in the pandemic, maybe February of 2020. Vincent Munster called me up and said that he was going to be infecting 6 rhesus macaques with SARS-2, and do I want some blood to do transcriptomics on? So I said yes. Like, we all were trying to get as many samples as we could back in those early days. This is a model of moderate COVID. These animals don't ever meet endpoint criteria, but they do have visible lesions on their lungs. They have microscopic lesions as well on histopathology, and they have infiltration of their lungs on X-ray. And then these are their whole blood transcriptomic signatures. It's very strange why on day 3 we don't have much of a signal at all, but on days 1 and 5 we do. And this was really interesting to me at the time, because Ben Tenover's lab had put out this study where they infected A549 cells with COVID, or with SARS-2, and did transcriptomics. So it was very clear in that interferon signaling is really suppressed. In this model, however, systemically, it's not. And it's totally upregulated on day 1. So I developed this hypothesis that the tissues that are infected directly do have this suppressed interferon no matter what. But in patients who have mild or moderate disease, and this is reflected here in this clinical data, they have these early interferon responses, and they can control the infection systemically. If they have severe disease, as again we see with these patients, they have suppressed interferon responses systemically and uncontrolled inflammation and severe disease. So in order to test this, we actually need an animal model that has differential severity, much like the collaborative cross. So go figure. Alpha and Omicron are naturally mouse adapted thanks to this N501Y mutation in the spike protein. So we could infect them directly without needing to use a transgenic or a mouse-adapted virus. And my former postdoc, Zainab Mohamdi, did this work. She found, again, differential phenotypes. Others have found this too. Ralph Barrick's lab, for example, has done some work with the collaborative cross and SARS-2. And these are not as extreme as the Ebola phenotypes. They're weight changes. I think we would probably see more changes if we used aged mice, but that's a future study. There's also differences in viral RNA. There's potential to model all these different COVID outcomes as we screen more CC lines or potentially use other things like aged mice. But we also see sex biases in terms of the weight. This is with alpha, I believe, which is more pathogenic than Omicron. And again, not super extreme. None of the mice die. Some of these, there are significant differences, but you can't see it on this, and it just looks like a bunch of overlapping error bars. But we do see some pretty striking differences between males and females for some of these. So we're going to continue that. We also have just started doing some mpox work, and I got funded for this a couple years ago, but unfortunately, Shay, my student, and I are both foreign nationals in Canada, and it took us 2 years to get our security clearances renewed. So mpox is an SSBA, or a select agent in Canada, and we couldn't do the work until we were both cleared to do it. So now we're finally getting started with that, but in the meantime, we collaborated with Alison Kelvin at the University of Calgary to do these studies in the CAST model, which is one of the founders of the collaborative crossline. And what we found was decreased spermatogenesis. We also saw the Leydig cells, which produce testosterone, were starting to die. And it looked like inflammatory macrophages were probably responsible, or at least there were a lot of infiltrating ones. So it's a good start, and we think that because the CC uses CAST as a founder, and Bernie Moss had previously developed this model and actually linked susceptibility to disease to the interferon gamma gene in CAST, we can actually select collaborative crosslines that have that CAST interferon gamma and make those comparisons related to sex. So we're going to get started with that as soon as I get back to work, and I'm on medical leave right now, so it's actually being delayed a little bit longer, but we are going to get started with that. So now I'm just going to finish up, and I stupidly didn't bring a timer up here. I hope I'm not that far over. But I'm just going to finish up by talking about some of the AI work that we're doing. And I'm going to finish up actually with policy stuff, because I think that doing AI work is going to become much more and more important for anybody who wants to get funding, because there is such a de-emphasis on the lab protocols. But basically what our approach is to this is we take all of our transcriptomic data, we find different features of small profile. We usually do this by differential expression, and I'll show you how. And then we select the features that are going to be the most discriminatory, and then we use different machine learning classification models to try to predict outcome with this data. So we've done this with a number of different species, and I'm just going to show you the Ebola stuff today because it's the furthest along. But what we'd really like to do is start doing this across species so that we can make use of all the data that is in the public domain. So we've developed a pipeline where we actually ortholog map everything to the human genome, since it's the best annotated genome that's out there. And that way we can actually start doing animals and model systems that may not have a great reference genome to use, or may not be very comparable to humans. This allows us to work with any kind of non-human primate and all sorts of weird models that some viruses need. So this is how we selected the classifier profile. We, instead of doing differential expression the way we normally do it, which is relative to a mock or a control sample, in this one we looked at lethal versus tolerant differential expression directly. And that's because we wanted things that were really different from each other in the two different outcomes. We also picked only the ones that were present at all 3 days, because if you're going to turn this into some kind of diagnostic assay, you're not going to be asking, did you get infected with Ebola yesterday? Or did you get infected with Ebola 3 days ago? You're going to want something that works no matter what. So that's what we chose. And we did this, and we found, with random forest, which is the model that we used, we found almost perfect classification, so 99.5% predictive performance. We also looked at the one paper that has used this approach to try to predict outcome in human patients. This was from John Connor's lab and Miles Carroll's labs. They used random forest and Savort vector machines, which both of them outperformed CT value in terms of prediction. So we wanted to see if we could use this data set to actually validate the classifier that we chose based on our mouse data. We thought this was pretty good. It's not good enough to perform in the clinic, but we got 75% predictive performance after we mapped these to the human orthologs, trained the model on this, tested the model on this, and this is what we got. So we thought it was good proof of principle. It still outperformed CT. It was roughly in the neighborhood of what John's lab found. When we added that data from those cynomolgus macaques that I showed to the training set, also ortholog mapped to the human, we actually boosted this to almost 96%. So we thought this approach was pretty good, and that's why we've developed this pipeline to try to integrate species so that we could make conclusions that might be applicable to people using whatever model is going to work, because sometimes you don't always get a choice. So now the last thing I'm going to talk about that my lab is doing is this, and this is where vaccines come into it as well. So this is the model that we're developing called pathogenesis, which is the host-focused pathogenicity and gene expression prediction system. And boy, do I need help with branding. I just wanted something that was about pathogenesis. But anyways, this is our approach. So to have a training set that can discriminate multiple parameters from each other, you need to have really, really well annotated training set. This, for anybody who's ever looked at SRA metadata, you are not going to get that by looking at SRA metadata. It's oftentimes a mess. So this means that you have to go through every single sample and sometimes pull this information out of a paper. I tried to do this for one sample from a data set that I had never seen before, and it took me a full day, 8 hours to annotate the whole thing, just reading the paper. But now, AI, thank you, AI agents, we are in the process of training them to extract this metadata for us. We'll have a human-in-the-loop session so that we can make sure that the agents are doing their job correctly. But this should, if it works, speed this up significantly. Then on top of that, we're also doing differential expression on all these data sets, so that way we can select classifiers based on that. And then we put this all into the training set. This will allow us then to predict what pathogen is it, what route of infection is it, what's the tropism, what's the disease presentation, what's the severity, what's the mortality, and if we're talking about a vaccine, what's the protection, potentially what are the negative health effects as well. So I think this is something that I'm hoping will be viable, just because I'm not sure what I'm going to do if I'm not able to work in the containment lab all the time. And there is a wealth of data, transcriptomic data out there that can be used for this. So I think that this is kind of the future, and unfortunately and unfortunately, it seems like that's where a lot of the funding is going too. So this is the last part I'm going to talk about, and this is the policy part. Jim said I was a staunch online advocate for vaccines, and I am, but this is not going to be as spicy as my online content. I'm just going to talk about the different policies that would have affected my research. And so this is a presidential executive order from Donald Trump that came out last May, about a year ago, and it lays out these 8 different criteria for something that is quote unquote dangerous gain of function research. So that basically means almost all research, because these 8 criteria are so loosely written that they can be applied to anything, and they already have been for many people. It's being applied selectively. It's not really clear how some of these decisions are being made, but this is the way that it's one mechanism by which the US government is defunding the NIH and science altogether. So, for example, conferring to the agent or toxin resistance to a clinically or agriculturally useful prophylactic or therapeutic interventions against that agent or toxin, or facilitating their ability to evade detection methodologies, that outlaws molecular cloning. If you put in an antibiotic resistance marker, you have just violated that provision. And again, it's not being applied broadly yet, but it is, these 8 criteria are being used to justify defunding grants. That's because of this. And this was originally covid.gov, the website where you would go to get information about testing. And then this showed up. And as somebody who has authored multiple papers on the origins of SARS coronavirus 2, first of all, it wasn't a lab leak, probably. All of the evidence suggests that it's not. I did not understand why everybody was so upset about the lab leak and why there was such an effort on the part of the government to promote the lab leak. And then this came out and I suddenly understood. With that executive order that potentially bans almost all lab research, and the idea that the pandemic started in a lab funded by Tony Fauci, is justification for ending NIH funding. And I'm not saying this because I want to focus on the US. Obviously, I work in Canada, and I don't have much US funding right now, but it's going to affect every single country because so many people do have US funding that the loss of all of this funding is going to impact other countries tremendously. And the efforts to replace what is being lost at the US are insufficient, just in terms of the numbers. So that's what the lab leak is about. And I did not understand it. I did not understand why I went through 5 years of arguing with these people on Twitter constantly until I understood that it's going to be used to take everything away. And here's an example of when this has happened. So Chris Brook is at the University of Illinois. He studies seasonal flu strains, and he studies how they evolve. And this work was flagged as dangerous gain-of-function research, even though it's not particularly dangerous. It's seasonal flu strains that are older, so everybody probably has some level of immunity to them. And basically, it was used as an excuse to strip him of his funding for no real good reason, because it actually isn't very dangerous. But they suggested that by adding mutations or even selecting naturally for mutations in these seasonal flu strains, it was gain-of-function research, which I don't agree with. But this is also the key thing. I fear a huge swath of grants will now be paused because of this uncertainty, because there aren't actually NIH guidelines for dangerous gain-of-function research. There's just this executive order. So NIH doesn't know what to do, and they're not funding any of these grants that might fit these criteria because they don't know how to do it. And it's being applied selectively for people who are working on vaccines. So this is a real problem, I think, for a lot of us, because if your grant gets noticed by the wrong person, you could lose your whole grant. Now, here's another thing that would affect anybody who's working on sex bias, and that are these 2 executive orders that came out on Inauguration Day last year, defending women from gender ideology extremism, restoring biological truth to the federal government, which doesn't really mean anything, and if you've read that executive order, I'm not sure that that's biological truth. But basically, this and ending radical and wasteful government DEI programs and preferencing both have been used to cut massive amounts of grants based on keyword searches. In fact, in the early days, the Doge people were using ChatGPT, and they were using like Control F to find words like trans. So transcription, translation, there's no rhyme or reason to it. And importantly, we've had a lot of discussion here about having places to talk to each other respectfully. And I think that's incredibly important, to go over all of these questions and to make sure that everybody is heard. But this is something else. I mean, this is cutting things just based on who a person is or the words that they're using. And this is absolutely the polar opposite of scientific. So yesterday when Greg was talking, sorry, Greg, caught you looking on your phone there. I'm just teasing. Greg said it best, that politicization of policy is bad. And I would say that it's not so much politicization, it's partisan behavior. This is not politics as usual, I think. This is discrimination. We who are interested in studying these sex biases and who've recognized that it is a gap, and on top of that, it's a gap that's based in gender discrimination and sexism. This is making it US policy. And that's hard because, and I know Sabra probably knows this too, writing that section in every grant about sex bias where you say the NIH has an initiative to make sure that we're looking at this, because there's too many studies that have been done with only male animals or only men. And this basically makes it so that we're going to go back to those days, and we won't be able to fund anything that at least says in the grant application that we are going to do otherwise. And this is another thing that has impacted me a lot as the co-editor-in-chief of Vaccine, is that the federal government right now is really cracking down. And I'm not defending Elsevier, okay? I am not, Elsevier is Vaccine's publisher, and they do make a lot of money. They make way too much money for the services that they offer. However, getting rid of scientific publishing altogether, which is the argument that is being made, I think is really bad. I think there should be alternatives, of course. There should be preprint servers. There should be lots of ways of disseminating information. But at the end of the day, the peer review process is really important. And eliminating that in journals helps justify eliminating that in grant making. And that is a real problem. There's not a lot of papers written about it, but I have spoken to people who have gotten, scores of 4 on their R01s. That's really good for anybody who hasn't applied for an R01 grant, not getting funded. And the reason for that is because after the study section, and they got rid of advisory councils in a lot of cases, they now have political appointees who are essentially screening these grants for political purity and compliance with these executive orders and other initiatives of the Trump administration. So this is going to be incredibly harmful to all of us too, because if nobody trusts the scientific publications, if the peer review process is completely disrupted, it's going to be impossible to get a grant based on its merits. It's all going to be partisan and political. And we've already seen this. This is a little bit old, actually, because this was all of the 2025 data. Max Kozlov published this in January. And this shows the number of newly funded grants at the NIH. And as you can see, it's going down. There's another graph that's out there. Yeah. There's another graph that's out there that shows that it's even lower now. All of the science funding agencies, all the science agencies in the US have plummeted in terms of their budgets. And then these are just the consequences of all of this stuff. We have out of control measles outbreaks in the US right now. In all of these little pockets. This is just what's gone on this year so far. We've almost overtaken what our measles cases were last year. We will definitely be losing elimination status. Hundreds of thousands of people have died because of the USAID cuts. We have had vaccines being descheduled. We have people not getting vaccinated as much. And then my friend, David Morans, he did try to conceal his emails from public records requests in a very stupid way, and I was a witness to this crime. I don't think that merits him facing 58 years in federal prison, however. He was arrested, he's 78 years old, he was arrested, cuffed, stripped, and perp walked, and his crime was basically saying, use my Gmail instead of my NIH email. So they are starting to arrest scientists. He was Tony Fauci's senior advisor, and that is basically what he's been arrested and charged with. And then just the last thing, the science that is being done, science is not gone from the Department of Health and Human Services, but it's changed in the sense that now there's millions and millions of dollars going to these initiatives that are not very clear or transparent. This one, however, this was reported yesterday by Christina Jewett at the New York Times, is basically to reinterrogate autism, reinterrogate thimerosal, reinterrogate all these things that have already been decided, essentially, or already been studied, not studying them in a new way, just trying to study them in a way that gives him the answer that he wants. So I think that this is a real problem for our research because this is where the rest of the funds are going. They are not going to actual researchers or vaccine research. And so that's just the end of my talk. I didn't want to get too into the weeds. I wanted to talk mostly about science, and I was so glad to do so. Thank you so much for having me.